Protein synthesis is the process of building proteins based off the information given from the DNA. Cells need protein synthesis because it builds proteins which catalyze necessary reactions in the body and make up specific parts that are necessary for life. In protein synthesis, the process starts with transcription, which happens in the nucleus of the cell. In the nucleus, there's DNA, which copies into pre-mRNA and then later becomes mRNA. And as mRNA leaves the nucleus into the cytoplasm, it enters a process of translation in which tRNAs build amino acid chains, which are called polypeptides. The polypeptides then form 3D structures, which are the proteins that make up these reactions.
Transcription is the process that copies genetic information from DNA to RNA. The goal of transcription is to make RNA, which will later be used to produce proteins. There are 3 steps of transcription: initiation, elongation, termination.
In initiation of transcription, the tau box is placed on the promoter and about 30 nucleotides from the starting point. These transcription factors land on the promoter and attract the RNA polymerase II. Once the RNA polymerase binds to the promoter, then it comes across and unwinds the DNA and creates the beginning of RNA synthesis. mRNA is synthesized on the template strand, and the other strand is the non-coding strand. Because RNA is only single-stranded, you have to make sure it is synthesized only on the template strand.
Elongation of transcription begins with the RNA polymerase II as it moves along the DNA strand, and this untwists the double helix, and this exposes 10 to 20 base pairs pairing with RNA nucleotides. The RNA polymerase reads the sense DNA strand from the 5 to 3 direction, adding nucleotides as it goes.
During termination of transcription, the pre-mRNA is released as RNA polymerase II reaches a stop codon or a terminator. As RNA polymerase II heads downstream, it eventually reaches the terminator, which is the sequence AAUAAA. And once it hits that, it continues 10 to 30 bases more and then releases itself as well as the pre-mRNA, which ends transcription.
mRNA processing is when the pre-mRNA gets a modified guanine cap and a poly A tail. A 5' cap has to be added to the pre-mRNA in order to protect from degradation and assist in ribosome binding during translation. The poly A tail must be added to the pre-mRNA in order to help protect from degradation, help transport the mature mRNA in the cytoplasm, and assist in binding proteins during initiation of translation. The parts of the pre-mRNA are the 5' cap, the leader region, the start codon, the parts of the coding parts of the mRNA, the stop codon, the trailer, and then the poly A tail.
mRNA splicing is a 2-step process in which the introns are removed from the pre-mRNA and the exons are joined together to form a new mature mRNA. An exon is a coding region that codes for amino acids, and an intron is a non-coding region that does not code for amino acids. The intron must be removed from the pre-mRNA before translation. The spliceosomes edit the mRNA, and spliceosomes contain snRNAs and other proteins. These snRNAs are also called SNRs. There are 2 SNRs at each end of an intron, which pull the intron together to create the exon.
The mRNA coding dictionary is used to determine which specific mRNA codon codes for each amino acid. So for example, if you do CGA, that codes for arginine, and AUG is the start codon, and that codes for methionine. And there are 3 different stop codons, and those are UAA, UAG, and UGA. The 3 stop codons do not code for a specific amino acid.
In translation, the mRNA attaches to a ribosome in the cytoplasm at its binding sites. At the binding sites, the mRNA attaches to a tRNA, and the amino acids form into a polypeptide chain, which become the proteins.
Ribosomes are the organelles where the binding between the tRNA anticodon and the mRNA codon actually occurs during protein synthesis. The ribosomes are composed of a large subunit and a small subunit, which are made up of ribosomal RNA. The ribosome is made by RNA polymerase I and other proteins in the nucleolus of the nucleus. Each ribosome has a binding site for the mRNA and then 3 binding sites for the tRNAs. The P site is the binding site where the tRNA with the growing peptide chain can stay, and the A site is where the tRNA with the new amino acid enters, and the E site is the binding site where the used up tRNA that's no longer needed can exit the ribosome.
RNA polymerase III makes tRNA molecules which consist of about 80 nucleotides which form a 3-dimensional L shape. It includes a loop containing the anticodon. It also has an attachment site at 3' end for the amino acid. tRNA molecules are made along genes in the nucleus by RNA polymerase III.
Amino acyl tRNA synthetase is located in the cytosol, and it's a special enzyme that connects each tRNA to its correct amino acid. There are 20 different forms of this special enzyme, and there's one for each amino acid.
In the initiation of translation, the mRNA attaches to the small ribosomal subunit at the start codon. Next, the initiator tRNA carrying the amino acid methionine and the anticodon UAC pairs to the AUG bases of the mRNA. This occurs at the P site, and then later the large ribosomal subunit attaches and completes the translation initiation complex. The initiation factors are required to bring all the components of translation together.
Elongation of translation is a 3-step process where amino acids are added to a growing protein chain. It begins with an amino acyl tRNA entering into the empty A site, where it then must match its anticodon with the correct codon in the mRNA in order to add the correct amino acid to the growing chain. In the P site, a polypeptide bond must occur between the polypeptide chain in the P site and the new amino acid from the A site. This is the step that separates the tRNA in the P site from the growing polypeptide chain and transfers it to the tRNA in the A site along with the new amino acid. During translocation, the tRNA in the P site now goes to the E site and exits, and the tRNA in the A site now moves to the P site, allowing the A site to be open for another tRNA to enter. Translocation ensures that the mRNA will be made in a 5' to 3' direction, and this 3-step process will continue to occur until the polypeptide chain is done, and it only takes 1/10 of a second.
Termination of translation occurs when a ribosome reaches a stop codon. The stop codon is a nucleotide triplet and the messenger RNA that signals the termination of translation. There are 3 different types of stop codons: UAG, UAA, and UGA. At the stop codon, the release factors connect to the mRNA and seal the polypeptide chains. The polypeptide synthesis is repeated until the stop codon is reached, and everything is then reused.
For amino acid structure, there's the amino group, the carboxyl group, the central carbon, the hydrogen, and the R group determines which amino acid it is out of the 20, and there are 20 amino acids total.
A peptide bond forms through the process of dehydration when the carboxyl group of one amino acid reacts with the amino group of another. This causes the OH from the carboxyl group and the H of the amino group to react and causes a water to release. thus forming a covalent bond between two amino acids, also known as a peptide bond. This process takes place in the large ribosomal subunit and is necessary to create a protein.
Polyribosomes are a cluster of ribosomes that are attached to one strand of mRNA. This occurs while a mRNA is undergoing translation and multiple ribosomes attach. This makes protein synthesis occur faster.
There are 4 levels of protein structure. There's the primary structure, the secondary structure, tertiary structure, and the quaternary structure. The primary structure is the actual sequence of amino acids determined by DNA. The secondary structure is the sequence of amino acids from the primary structure twisted and held together by hydrogen bonds in the amino acid. The tertiary structure is a combination of all of the secondary structures. They are held together through their interactions in electrical charges between the R groups in the surrounding waters. Finally, the quaternary structure is a combination of proteins from the tertiary structure to create one big cluster of proteins.
Modifications in the Golgi apparatus occur, and these deal with phosphates, lipids, and sugars. And different amino acids could be added to the polypeptide chains or taken out. Proteins are modified to maintain their three-dimensional shape, and these modifications help make the protein fulfill their purpose.